Hello, and thank you for joining us today for the webinar entitled Role of Biobanks Challenges and Opportunities in Cancer Research. I'm Shonali Paul, the COO of CloudLIMS.com. I'm here to moderate this event. We are pleased to have amongst us Dr. Birendra Yadav joining us today as your presenter. Dr. Yadav has a rich experience of 11 years in the area of cancer, stem cell biology, biobanking, and hematology. He is currently responsible for managing the Biorepository, Department of Research, Rajiv Gandhi Cancer Hospital and Research Center, India, as a scientist come technical manager. Dr. Yadav is a PhD from the Department of Anatomy and Cell Biology, College of Medicine, Kangwon National University, Republic of Korea. So, Birendra, a very warm welcome to you today. Before we get started, let me remind you that this is an interactive webinar. We encourage you to ask questions anytime during the presentation by entering them in the question box to your left and clicking send. The questions will be answered during the Q&A session right after the presentation. We will take as many questions as time allows. If we are unable to answer your questions today, we'll answer them offline. It is time now to hand over the floor to Birendra. It's over to you.
Thanks, Sonali, for an introduction which focuses where it makes me larger than life. I am humbled. Today I am going to share my views about role of biobanks, challenges and opportunities in cancer research. We will cover the following points in our presentation. Biospecimen biobanks, what they are, how they work, the objective of biobanks, current cancer scenario, cancer research opportunities, role of biobanks in cancer research, major challenges facing biobanks, and how to overcome the challenges. So, what are biospecimen banks? First of all, we should know about the biospecimens. So, what are human biospecimens? Biospecimens are samples of biological material such as urine, blood, tissue, cells, DNA, RNA, protein from humans. To store a biospecimen, we need biospecimen bank. So biospecimens are often stored in biorepository or biobanks and are used for laboratory research and biological purposes. Biospecimen banks are organized collection of human specimens, associated data and information stored in controlled facilities for an advancement of biotechnology, human health, research and discovery. Biobanks are one of the list of 10 ideas changing the world right now, published in Times magazine 2009. The article says that biobank is a safe house of tissue samples, tumor cells, DNA, and blood that should be used for research into new treatment of diseases. Biobanks are an important compound of personalized medicine and strongly support the scientific progress in stratification of population of biomarker discovery and validation due to progress in personalized medicine. Biobanks are an essential tools for new drug discoveries and development. It play an important role in whole process of patient prevention and prediction, follow-up, and therapy monitoring and optimization. Biobank have the specificity in that they cover multidisciplinary approaches to the human health. And these approaches are combination of biology, medical approaches, bioinformatics technologies, computationing, and modeling. So generally, we can say that biobank consists of three groups of relatively distinct information. The first is biological human material. The second is attached or connected information. And the third is the legal issue like consent and patient or individual data safety and protection. And this makes the science of biobank so comprehensive and complicated because it requires a multidisciplinary and universal approach in all stages of life cycle of the biobank. Types of biobank. Classification of biobanks is not simple and is based on different approaches. Taking into consideration the biobank's nature, purposes, scopes, biobank can be classified into these measures groups. Based upon the sampling, it can divide into two disease-oriented biobanks and population-based biobanks. And population-based biobanks, some biosamples are projects links genetic data with the information of health status, lifestyle, and environmental factors. While in disease-oriented biobanks, samples are collected from the patients and aiming at discovery and validation of genetic and non-genetic risk factor of disease. Based upon the business model, that way there is a commercial biobanks and non-commercial biobanks. The best example of the commercial biobank is the stem cell biobank, where donors storing their stem cells for their future use. And for this, they are paying to the biobank. Non-commercial biobanks, where biosamples are stored without any charge from the patient, and those samples are provided for the research purposes. Based upon the funding, that who is funding to the biobanks, whether it is the public funded or private funded. And operation scale, whether that biobanks are sample of storage, sample are stored in single laboratories or biosamples are stored from the whole hospital at one place, or number of biobanks are interconnected, making network to store their samples at one place, whether it is a national biobanks or regional biobanks. So depend upon the scale of biobanks, there are also many types of biobanks. So in the small whole biobanks, in lab biobanks, investigators are storing their samples, storing the samples for their project. So if you will see the current types of biobanks or classification of biobanks, so according to the BBMRI, European Biobanking and Biomolecular Resource Research Infrastructure, there are only two types of biobanks. Population-based biobanks, where biobanks focused on the study of the development of common complex diseases over time. And the disease-oriented biobanks, where tissue samples and clinical data are used for the clinical applications or disease orientation and disease. So biobanks in the Asia Pacific region. Major tumor biobanks are located in developed countries. The countries like the US and Europe have the largest market size in biobanks. Europe is the largest biorepository market due to the huge demand from Netherlands, Italy, Germany, UK, and several other countries. However, the rapid growth in population in Asia Pacific countries will affect the market of the developed countries. The large population and vast patient pool in the region will drive the market for biobanking in Asia Pacific region. Here you can see there's some popular biobank in Asia. Rajiv Gandhi Cancer Institute Research Center Biobank. It is the first biobank in North India. Other popular biobank in India is Tata Memorial Cancer Center Biobank, which is also the oldest. Few biobanks are also under the construction in India, and the largest means future largest biobanks are under the construction at National Cancer Institute, Jhajjar, Haryana. So, biobanks in some other countries, in China, that is a cancer center tissue bank, Funan University, which is popular for the blood, tumor tissue, and body fluids. Japanese collection of bioresearch material, popular for the storage of animal, plant tissue, and microbes. Singapore National University Tissue Repository, popular for the human cancer tissue, breast, liver, bone, and it is also storing some other types of biosamples. Israel National Laboratory for the Genetics of Israeli Population, DNA, human cell lines. One another are popular biobanks in Singapore is SingHealth Tissue Biorepository. So many researchers use biospecimens. So major tumor biobanks are located in developed countries. Researchers ask for many types of biosamples for their research. Most biobanks, tissue banks, and biorepository collect blood and its component like whole blood, plasma, serum, RBC, and white blood cell. Other biosamples collected at biobanks are buccal swab, DNA, RNA, protein, cell lines, fluid, urine, cerebrospinal fluid, synovial fluid, amniotic fluid, buffy coat, bone marrow, stem cells, and tissue. Researchers use these samples to understand progression of disease or to understand the response to certain treatment. To measure drug concentration, proteomic, metabolomics, and epigenomic study. And researchers also use to measure the drug concentration, to measure sugar level, antigen-antibody interaction in allergic reaction. So different type of samples has the different type of use for the researchers. How specimen banks work? So the first step in the biobanks is approval of ethical committee. And most important things is informed consent should be taken before procurement of biosamples. If somebody collect biosamples without informing and taking consent from the donor, it is unethical. And most of the researchers in the past and also some researchers in the present are doing and collecting the samples for their research from other hospitals without the taking the consent and informing into the patients about their sample and its use. But it is unethical. So after obtaining the informed consent, biosamples are collected with the help of doctors, nurses, and pathologists. Pathologists are well trained to distinguish about the tumors and normal tissue. And if the tumor here are less than 2 centimeter, is not stored for biobanking because diagnosis of patient are more important. So it is important for the biobanking that we should give first preference to the patient for their diagnosis. If the sample are more than 2 centimeter, then we are going for the storage in the biobank. So confirmed tumor, adjacent normal tissue with other biosamples like blood, urine, plasma, serum are stored at biobanks at in liquid nitrogen at minus 196 or at minus 80 degrees Celsius. Formulate-based blocks are also made for quality control or also for some research purposes. After so associated information like pathological, demographical, social, and follow-up data are collected and linked with the stored samples. The most this also it is important for the biobanking is the time of storage. Samples should be stored within 30 minutes. 30 minutes because once the biosamples are detached from the body, the hypoxia condition is generated in the biosamples, and there are a lot of genes are going to express or and other proteins and biomolecules are start to degrade. So it is important that samples should be stored within 30 minutes or before the 30 minutes. And second thing that those biosamples are stored in the biobanks are de-identified with some unique identification number so that researchers don't researchers cannot identify or identify the patients with unique identification number. So this is also the point of ethical to de-identifying the patient according to the HIPAA rules. And once we receive the request for the biosample projects, uh, so the project are evaluated by the biobank manager, and then it is tissue release board. Release board is seeing the scientific and ethical point of view, and once approved by the tissue release board, sample are provided to the researchers for their research purposes. So these are the points. These are the point, and these are the steps biospecimen biobanks work. So how biospecimen bank affect cancer research? So biospecimens are critical to cancer research because they contain an extraordinary amount of biological information written in the language of cells, genes, and proteins that can identify the biological characteristic of cancer cells over time. Researchers can correlate these patterns with the clinical pictures of how different patients experience progression of their disease or how the disease respond to therapy delivered. Critical to making progress in our fight against cancer is the availability of high quality biospecimen. So if we will provide high quality biospecimen for the research purpose or for the diagnosis purpose, we will get good results. And a collection of the high quality biospecimen depend upon standardized handling process, removal of competitive barriers to biospecimen access, and biospecimen research science that explore how collection, processing, storage, and transport procedure impact the biospecimen's molecular characteristic and ultimate usefulness in cancer research. Because the current biobanking system has fallen short in all these areas due to the many challenges. And also, their banking system are not more popular, or researchers are not more aware about the biobanks. So, there is a considerable and problematic shortage in the availability of high quality, well documented biospecimens for cancer research. As a result, the progress and pace of research is hindered as researchers often question the quality of biospecimens available to them, and therefore the reliability of their research results. Scientists are compelled to restrict the scope of or curtail their project due to limited availability of biospecimen fit to their research purposes. This issue must be addressed urgently in order to enable advances in cancer research and patient care. The objective of biobanks. So, biobanks have some um, objectives. First one is to combine biological and medical approaches for efficient clinical investigation for patient health care. To facilitate progress in human health, including treatment for the most common genetic disorders. Uh, Suppose some genetic diseases are quite common and affect large number of people. For example, some form of cancer. So research is carried out into how to treat and cure these common type of diseases. Stem cell research, which is also important things for the human health. Then stem cell possesses significant potential for the treatment and curing of a wide range of genetic conditions, and some people may wish for their stem cells to be stored in case they need to use them in the future as a treatment. Further research is needed to unlock the full potential of stem cell health survey. So also, researchers need to understand better how genetic diseases affect people as their illness progresses. To be able to develop treatment depends on carrying out research with comprehensive collections of DNA samples collected from those affected by specific disorders. Environmental factors. So there are many diseases are generated due to the environmental factors. So each person's each person has their different genetic makeup and different type of single nucleotide from vulvar fission. So environmental factors are nowadays working as a to triggers to generate many types of diseases like asthma, cancer, diabetes, etc. So it is important to examine how things like air pollution, use of alcohol, tobacco affect the development of disease in different individuals, and also how these things are interact with genetics. So these data can be found in the biobank. Age demographic database. So many countries have an aging population where a greater portion of society is elderly. Healthy aging is an important area of research to improve the life of elderly people. And they are economically inactive, uh, and also they are heavy user of healthcare. And most important things of objective of biobanks are personalized medicine. So this is the treatment that is tailored to the particular genetic makeup of the individual receiving it. Humans are all similar, but there is a lot of genetic variation between individuals. So, as we were doing in past, that single medicine we are providing to all person affected from single disease. But treatment that work well for one person may not work so well for somebody else. So further research is needed how to develop personalized medicine. So biobanking is providing high quality biosamples for next generation sequencing and other diagnosis purpose on the basis of which clinicians are treating to the patients. So that is saving both life, money and time for the patient. Current cancer scenario. So cancer is a leading cause of death in both more and less economically developed countries. The burden is expected to grow worldwide due to the growth and aging of the population, particularly in less developed countries in which about 82% of the world population resides. Country like India, cancer is increasing rapidly due to the urbanization, westernization, and due to their food habits and environmental pollution. Here you can see the cancer incidence and mortality in some Asian countries. As per Indian population census data, the rate of mortality due to cancer in India is high and alarming. Cancer is the second most common disease in India, responsible for maximum mortality. Major causes of cancer. Cancer. So the occurrence of cancer is increasing because of the growth of aging of the population as well as an increasing prevalence of established risk factors such as smoking, lifestyle factor, overweight, physical inactivity. Also, due to the changing reproductive patterns associated with urbanization and economic development. Tobacco is the single most important identified risk factor for cancer. Due to the urbanization, there are a lot of the environmental pollution. There are also due to the pollution in the water. So heavy metals we are using in our vegetables, food, that is also responsible for the generation of the cancer. And so it is important to know that only 5 to 10% of all cancers are due to an inheritable gene defects. So we can't say that all cancer are due to the inherited genes. So remaining 9 to 95% cancer occur due to environmental defects, due to our lifestyle factor, due to our infections, due to lack of physical activity. So cancer research opportunities. So biobanks are imperative for progress in cancer research. Focuses on genetic variation and complexity among different caste, races, age groups by single nucleotide polymorphism discovery. Indian population harbor large number of genetic variation, and genetic variation have not been surveyed adequately at the continental level. The Indian continental group has significantly higher nucleotide diversity than other European and East Asian groups. According to some research papers, genetic diversity also varies from one caste or community to other. But some castes like Erula, Mala, Vadiga, Yadav has the highest number of genetic variation in comparison to other higher castes like Brahmin or Jain. Some population in India have highest diversity in the world. Novel DNA reservoir and biomarker studies is also the good opportunities for the cancer research. So most of the population are not used for research. So they have novel DNA reservoir for biomarker studies, which is possible with the help of Biobank. And most of population are not touched due to the inability of the biobank. Our sample are not possible to collect for their research. Due to the complex demographic history, unique social structure, and other lifestyle factors, cancer research opportunities also are effect of different environmental exposure to various type of chemical and radiation, and disease based on complex genetic background. So Asian countries like India, China and Japan has more opportunities for biobanking and cancer research due to their diversity. So if I will talk about India, it is considered as a treasure for the genetists and evolutionary scholars for research because of tumor heterogeneity. Different places have different type of cancer. Some of the cancer incidence are highest in the world. For example, gallbladder cancer is highest in Delhi. Some other cancer types like head and neck cancer are highest in northeast region, are found in India. Also, cohort studies is possible due to the highest number of second populous country in the world. India also has the highest human diet, genetic diversity in the world, complex genetic background and large number of diseases, and Indian population genetic variation has not been surveyed yet. Role of biobank in cancer research. So biobanks play a key role in providing access to quality cancer biospecimen and associated data, minimizing time between research and clinical application. Discovery of novel drugs, treatment therapies, and prevention strategies. Biobanks also play a role in allowing researchers to discover genetic association of complex disease, ensuring that new drugs may effectively reach out to patients. Biobanks also play a key role in the personalized medicine, and also the clinical diagnosis of the patient. So these are the things about the biobanks. But biobanks has also the major challenges. So major challenges facing a biobanks are lack of awareness, large capital outlay, sinking budgets, biobank data management issue, data sharing and security concerns, lack of awareness. So most of the researchers are not aware about the biobank and its importance. Just few months before, I did one survey among the researchers, professors and PhD students from different institutes, universities and research centers about the biobank to know that how much they are aware about the biobank. And I was surprised to know that only few researchers, professors or scientists know about the biobank. And they are approaching to hospitals or doctors for free samples. They don't know the importance of informed consent, and they are procuring the biosamples from the hospital without following any standard operating procedure, without without thinking about the time storage of the biobanks, and also the informed consent. So due to this, the samples which they are using are of not good quality samples. So the results which they are generating through the experiment are also not good. So due to the lack of awareness, also there is a delay in clinical investigation and patient diagnosis due to the unavailability of high quality sample. So if in our country researchers are educated or higher educated generation don't know about the biobank, how we can expect that patient or donor would be more aware about the biobanks. So, as expected, that patient or donors are not more aware about the biobank. So this is the challenges. Large capital outlay and shrinking budget. So biobanking requires money for new infrastructure, support personnel, operation of tissue banking itself, and quality control. So support for biobanking through government is not available in country like India. More people are not more aware about the importance of biobank. So philanthropic sources of support are also not more popular because of lack of awareness. And the, there are most of budget in our country. We have a lot of challenges. Suppose that challenges of electricity. Most of liquid nitrogen factories are established near the metropolitan city. Infrastructure are not good, so transportation are expensive. Accessories which are used in biobank are also expensive. So major portion of allotted fiscal budget is dedicated to establish and maintain biospecimen storage facility with uninterrupted electricity. And due to this reason, there are minimum scope for adopting any automation technology over manual method and for record and capturing information. So in some Asian countries, governments came forward. So biobanks of Korea, Japan, China have been rapid growth in size over the last decade due to substantial investment from governments. So biobank data management issue. So maintenance of good quality sample along with well documented sample, metadata and donor information are very difficult without data management software. Availability of an affordable IT solution that facilitate accreditation and fulfill audit requirement are not possible due to the shrinking budget, and it is expensive. There is also the whole requirement of the space and some 10% to generate information system. So, due to these reasons, regulatory compliances spreadsheets of error-prone, traditional on-premise LIMS applications are expensive. Sharing voluminous data, sharing voluminous data across multiple sites while maintaining data confidentiality still remain a daunting task. Data sharing and security concern. So adherence to strict compliance regulation. So if you have the biobanks, you have to adhere to some the strict compliance regulation. So that is the tracking chain of custody, enforcing electronic signature, audit trail maintenance, masking confidential patient information according to the HIPAA rule, and also there is requirement of some accreditation like CLIA, STR. There are also some challenges about the data security and integrity concerns. And that is advanced encryption standard, which is not possible when you are using paper and pen to collection of the samples and recording their information. Auto scaling facility is not possible, not easily possible in-house LIMS software. Automatic load balancing, daily backups, and disaster recovery. So when we will use the traditional way to for the recording and everything, so we will. It is not possible for these things, and also disaster recovery are impossible. So these are the challenges which we have in our biobanks. So how to overcome these challenges? So first important thing is to make a next generation biobanking and to accelerate research for tomorrow cure. It is indispensable to overcome from challenges. And these challenges are, and how to overcome with these challenges? That is need for awareness and need for automation. So need for awareness. How to make aware about to the researchers and also to donors. So there are many ways to make aware to researchers and donors by organizing the conferences, workshops, by email, and good journals can play a key role for the awareness about the biobanks by making a strict rule that about that only those papers will be published which use the sample as per standard operating procedure. We we can also make aware to the donors and encourage patient to donate biospecimens with the informed consent. Need for automation. So Biobank need to automate using cloud technology to achieve. We are saying the cloud technology because it is fast. Efficiency, it is fast in accountability, establish workflow standard, adhere to regulatory guidelines, and ethically share biospecimen and patient clinical information. So biobanks, a CloudLIMS person are more aware about the rule and regulation of the biobanks, and they adhere to the regulatory guidelines. So there are no need for affordable IT person at hospital, biobank, and server for the LIMS software when you use the cloud-based tools for biobank automation. And it is easy, it is quickly deployable, scalable, that when your requirement is increased, it can easily increase. Work efficiency is also increased. There are less chance of mistakes, secure and compliant, and cost effective. Thanks. Questions are welcome.
So thank you very much, Birendra, for the presentation. I'm sure all will agree that it was insightful and presented some very useful data. We have a number of interesting questions, so I'll move straight on to these. So the first question I have is, we are planning to establish a small-sized biobank in India for storing disease-based samples. What approvals do they have to get, and which bodies' guidelines do they have to follow?
First of all, I let you know that there is no specific guidelines in India for the biobanks. And ICMR defines biobanks in 2006, and they have the simple rule and regulation. So we are following that one. And at the international level, we are following the ISBA guidelines for that one. So there is no need for the pre-approval, and just you have to take the ethical approval for the consent and other documents to establish a biobank.
Okay, so then the second question is, then how does the ethical approval and the funding for biobanks work in India?
If you talk about the funding is very bad. That, as I told you, that government is not aware about the biobanks. They don't have any specific fund for biobank. So it is very difficult to run a biobank in India. And even the other person and our philanthropic, he can also not coming forward to participate in biobanking, to establish a biobanking, giving the fund to establish a biobanking. So funding is not good in India, and it is very difficult to run a biobank.
Okay, then the third question I have is, what will be the control sample for a cancer sample to compare some gene amplification?
Also, control samples that sometimes we are collecting the tumor cells and the adjacent normal cells. So at the same times, you can compare your research result from the same patients with that what is happening in normal cells, and you can compare with their disease cells, like tumor cells. So this will be the control.
So are normal samples readily available as control samples while studying cancer as a disease?
Yes, no environment.
And are consents readily available for those normal samples, or that needs to be provided while talking to patients and they need to be enrolled?
Yes, before the taking the samples from the patients, we are going to the patient. We are giving the information and about how those projects for which we are collecting the samples, and if those donors agree for giving a sample, and after the signing the consent, we are able to take a sample. So it is not possible to collect a sample from all patients or all.
The next question I have is, what is the possibility of setting up a population biobank in India that collects blood and urine samples and baseline phenotypic data?
Yeah, in India, there are a lot of possibility, and I want to say that there is no specific rule and regulation. So it is at present it is very easy to set up a population biobank, and but your documents should be ethically approved, and you have to convince to the general population about the biobanks, and if they are agree and they are signing the consent, then you can collect those samples.
So where do you get the approval for setting up the population biobank?
So as I told you that there is no need for the approval, but for your consent should be approved, ethically approved. So either you have your own ethical committee, or you have to consult her to the other organization where there is an ethical committee to approve your consent. So after the getting the approval of your documents, you can store. And I want to say that there are some guidelines still pending in our parliament. That first in 2006 to 2007, the Anthropological Survey of India convened a group of experts to develop a model law for the establishment of biobanks and how to run the biobank. And that draft is made and that is submitted to our parliament. But I don't know the current status because no one is talking about that one. So if that will pass, then India will have the strict rule and regulation for the biobank. And in the similar way, ICMR have also submitted one draft to the Ministry of Health for the biobanks guidelines, and that is also the pending.
So, how many samples do you currently have in your biobank, and how are you managing the data in your biobank?
So if you talk about the frozen samples, so we have the 10,000 frozen samples and more than 40,000 paraffin blocks. And initially we were doing on the paper and pen work, but when our sample size increased, then we approached to our IT because we have the big IT department, but it was very difficult to explain them about the biological things and how to collect the samples, means clinical data, pathological data. So they were not very trained to set up a software for our biobanks. So at that time we approached the CloudLIMS, and those persons are well aware and well trained about this one. So we have the CloudLIMS software and we are managing everything through Cloud.
And with regards to consents, what guidelines are you following, and are they the same for creating cell lines as well?
Yeah, I think actually that is the same guidelines for the cell lines and biobanks, and we are following the ICMR guidelines. ICMR has the clear guidelines to how to take the consent and what information should be, and your consent should be in bilingual language for those donors from which you are going to take the sample. That consent should be in his or her own language. So we are following the ICMR guidelines for the consent.
And do you also have people educating the donors for the approvals and the consents that they're making? Because that also needs some education, as your presentation said, that some conferences and workshops need to be handled, as well as there need to be somebody within the facility who explains the importance of donations as well as the reason for taking consents to donors.
Yes, we have one person that person is going to the patient and explaining about the biobanks and its importance. So sometimes yeah, people become happy that they, those samples which they have already dissected from the OT. So they are happy to know that their sample will be used for the research and some other person will be benefited from their own sample. So uh, we have one trained person, and he's giving the more information about the biobank.
Then, other than are the ICMR guidelines published on their website, and is there any other body other than ICMR that biobanks need to follow?
No, there is no other guidelines except the SMRs. But generally, biobanks in India, like our biobanks, we are following the ISBAR guidelines, and that is the
Only
biggest
ICMR
organization.
guidelines then.
Yeah, so only ISMR guidelines available in India at present. So as I told you, that two guidelines are still pending in our parliament. When we will get the approval, so we will have the clear guidelines for the biobanking.
Then, with regards to the SOPs that you follow for cancer tissue processing, are there any standard protocols? For example, how soon do you get the samples into the biobank to ensure that the quality is not compromised? What should be the process for dissecting the tissue after upon receiving it from an operative procedure, so on and so forth?
Uh, yeah, so it is very important for biobank to collect the samples and store as soon as possible. And as per the ISBAR guidelines, samples should be collected within 30 minutes. And we are lucky that our means OT and pathology department and biobanks are connected, not so far distance, and that our one person is already waiting for the sample in front of the operation theater, and as soon as sample is resected from there, the same person is bringing the sample to the pathology department, and pathologist is doing the grossing for the sample and giving the tumor tissue, normal tissue to the biobank person, and we are de-identifying the samples, labeling the samples, and we are storing the samples in the liquid nitrogen at minus 196, and whole process we are storing the sample within 30 minutes.
Okay. So unfortunately, this is all that we have time for today. So any questions we could not take today, we will answer them as soon as possible. Thank you for your time, everybody, and have a good day.
Okay,
Bye-bye
thank you,
from
thank you.
cloudlims.com and from Dr. Yadav as well.